Hi everyone.
Hi.
Now we biology the second chapter and so let's begin this class. First of all, we start with what is cell division. Cell division is one of the fundamental characteristics of life. It enables life to perpetuate generation after generation. Now next is structure of chromosome. Nucleus contain most of the cell DNA and are organized into discrete units called chromosome. It contains DNA and a protein. This complex mixture of DNA and a protein is called chromosome. When the cell is divided, it appears to be thick and condensed form. When the cell is not dividing, it appears to be thin dark chain fiber. So this is called chromosome. This chromosome readily pick up dyes and get color. So that is why it is called chromosome. That means colored body. So chromosome is highly condensed and called chromatin fiber. So discovery of chromosome. Chromosome was first discovered in 1882 by a German scientist, Walter Fleming. He discovered it by using the larvae of a salamander. He discovered it from using his old microscope. He discovered that a substance arranged on the basis of height. He called it as mitosis. So later new discoveries were found on the basis of new development of microscope. So next is chromatin material. Chromatin material is a fiber constitute which is made up of two substances. DNA, that is deoxyribonucleic acid, it contains 40%, and a histone, a particular kind of protein, that is 60%. So next is molecular structure of DNA. Molecular structure of DNA was first discovered by Rosalind Franklin in the year 1952. Later that Watson and Crick discovered that DNA was a double helical structure. So they get Nobel Prize in the year 1962. So a single DNA molecule is very large and it is called a macromolecule. It is composed of two complementary strands wound around each other in a double helix. Each single DNA molecule is made up of repeating nucleotide made up of three components. Sugar, phosphate and a nitrogenous base which extend the sugar inward to join the nitrogenous base forming a ladder-like or rung-like arrangement. So, four nitrogenous bases are found in DNA: adenine, guanine, cytosine and thymine. Adenine pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. So, next is histone. Histone is a protein that helps in coiling and packaging DNA into structural unit called chromosome. A single DNA molecule wound around 8 histone protein. This complex mix is also called a nucleosome. A single nucleosome is observed like a football. That is, a football is wounded with 2 or more lobes. So, our DNA molecule is 2 meter long. Our nucleus is 6 micrometer long. So, the DNA has to be highly condensed to fix inside the 6 micrometer long nucleus. So it is highly condensed as like an electric cord, or we can observe it as a telephone cord. So next is structure of chromosome. Each chromosome in its condensed form is visible when they start cell division. Consists of 2 sister chromatids. Each sister chromatid is joined at some point called centromere. This centromere is located at a particular site. This centromere also serves to attach spindle fiber during cell division. As the spindle fiber contract, the 2 sister chromatids move to each pole, and after the cell division, they grow as their parent cell and they move to their original position. So next is genes. Genes are special sequence of nucleotide of a chromosome that encode a particular protein that express some particular feature in the body. They are the unit of heredity which transform the body character or a parental character from parents to offspring. So they are the unit of hereditary. So there is a question that it's a general knowledge, that is, who is the father of genetics? Father of genetics is Gregor Mendel. So next is, what is the need of division of cell? Cell division is essential for 4 things: growth, repair, replacement and reproduction. So we come to the first, that is growth. All organism, including ourselves, is made up from a single cell, and it divides into cluster of cell and starts shaping into different tissues and organs. So cell is essential for growth. So next is replacement. There is always a wear and tear of the cell. That is, for example, 2 million RBC from our body are replacing every day. So cell is essential for replacement. So next is repair. Apart from normal wear and tear, there is always a wound in the skin or a broken in the joints. So cell is essential for repairing the part of bones and closing the wound of the skin by growing the new cell. So next is the last is reproduction. So cell division is essential for reproduction. For example, there are two types of reproduction, that is mitosis and meiosis. In mitosis, the parent cell divide into two daughter cells. And only the parent cell, there is no sexual interaction between the parent cell, that is it divide into two daughter cells. So this is also called the interphase. So next is in meiosis. It is a sexual reproduction, that is a sexual interaction between sperm and ova. So in this process, it is only active in reproductive. So in mitosis is also used for growth and development, that is a cell. For example, amoeba, a single cell organism, is reproduced through mitosis. In meiosis, human, plants, etc. reproduce meiosis process. So this half portion is completed. So remaining portion we study in the next class. Thank you.